A very good morning to all of you. I welcome you all to the fifth and the last day of the National Training Workshop on Atmospheric Aerosol Instrumentation, that is NT-AAI. Today we have two lectures followed by assessment test, some instrument demonstrations, and finally the validatory function. Today's first lecture on bioaerosol characterization will be delivered by Dr. Sachin Gunthe, professor at IIT Madras and former head of the Max Planck Partner Group on bioaerosol research. He is also affiliated to School of Engineering and Applied Sciences, Harvard University, and Max Planck Institute for Chemistry, Mainz, Germany, as associate researcher. He has an experience of teaching diverse courses in the field of environment engineering and earth science, system, and climate change at the undergraduate and graduate levels. He was awarded the Fulbright Nehru Academic and Professional Excellence Fellowship, Fulbright Fellow, Harvard University, USA in 2016. He co-edited journals like Atmospheric Chemistry and Physics and Journal of Atmospheric Chemistry. His area of research mainly focuses on aerosol biosphere climate interaction, bioaerosol diversity, and ecosystem health impacts over India, and atmospheric chemistry climate modeling. With this short introduction, I invite Dr. Sachin Gunthe on stage to deliver his lecture. Please, sir.
Okay. Good morning, everyone. First of all, thanks to the organizers for having me here and a very short introduction which you gave. That introduction would have been, it's really incomplete without mentioning that I did my PhD at this very place, right? So it's a very special thing for me to be here with such a short introduction at a place where you did your PhD. So that's, again, thanks for the organizers to get me here. What I'm going to do, so what is the time that I finish at 10:15 or I take 45 minutes? You take 45. I take 45. Otherwise, Shweta ma'am will stand and she said that, wait, 10:15 now I start. Right? She's a strict teacher, by the way. What I'm going to do is that I'm going to take you through the different characterization techniques which are used primarily for the bioaerosol characterization. And then to give you an emphasis that how there is no standardized method to characterize the bioaerosol and what your requirement is. I'll also try to show you some results which are, can you hear me? Yes. We'll try to show you some results that how one particular results can be obtained by using a specific technique and can be supplemented with this particular. This is just as an outline for the today's talk I'm going to follow. I'll run through some introduction and motivation, then bioaerosols, the basic definitions, because a lot of people still do have some sort of a confusion when it comes to the bioaerosols. Why they are different from the other aerosols, like, whatever you have studied over the last 3 days. This is totally different. So more of a microbiology class than anything else. So don't mind about it. Why study the bioaerosol? Because of course, as like any other aerosols, they also do have the climate impact, and then they also impact the ecosystem in general, but that's in a very different way than what your other aerosols they do. Then, of course, the topic of today's talk, like, the measurement techniques. What are the different measurement techniques you use to characterize these bioaerosols, which is offline techniques, advantages and disadvantages. Then I have something about online techniques, disadvantages and advantages. You might have noticed that why I swapped these words between advantages and disadvantages. And then I'll try to clarify as I move along how to find a suitable technique for study objective. When you want to understand a bit more about the bioaerosols, what is the best technique you want to find out, which is like more suitable for your objective. And then I would just wrap up with summary and future scope about this. So let's get started with this. I always try to show this slide dedicated to Professor Paul Crutzen. I had an opportunity to be associated with it, that we are currently living in an epoch which is not governed by the natural activities, but we as a human are now kind of shaping the Earth's current behavior, right? So there is a lot of things which is going on, like, that this kind of exponential increase in everything. And then if you look into the different epochs, we are now probably here. And then this is the term which Professor Paul has coined, like, Anthropocene, that one of the epochs that which is more governed by the anthropogenic activities and not by the natural activities. So we have to be a bit of a responsible human being as far as the Earth's climate is concerned. Why they are different from the other aerosols, right? So if you look into here, unlike the gas phase measurements, that working in the aerosol field poses one unique challenge, that we run across the 5 order of magnitude size range, right? So anything which is greater than 1 nanometer and which can run up to around 100 microns, they are called aerosol particles. And then anything which is above 100 micron, kind of like cannot call aerosol, because they will very quickly settle down on the Earth's surface. And then not for a very long time, they would govern by the different laws, which basically governs that how a particle is basically floating around in the air. Now, if you look into the different type, of course, I would like to bring in your attention to these constituents, which are mentioned here in the green color, like the proteins, viruses, bacteria, spores, and pollens, that they also do have a size range from where they vary. And then my today's discussion would be more focused on these green constituents, and these constitutes primarily the definition of what do you call as a biological aerosol or the bioaerosol. Now, of course, like any other aerosols, you have the natural and anthropogenic sources. And then as I will try to convince you that not only from the natural, but then of course the bioaerosols also do have the anthropogenic sources. Like then they have the primary emissions, which is emitted from the biota. And then there is a secondary formation. I would not call it as a secondary formation, but it is more related with the transformation and aging of the bioaerosol. Of course, then they do take part into the cloud processing, because you know that the clouds cannot be formed without the presence of aerosols. And then it is true for the bioaerosol, except that the process through which they form the cloud or initiate the cloud formation is slightly different than what the other aerosols they do. And then the ones those who do not take the part in the cloud processing are, of course, washed out, deposited, and that's called as the dry deposition. And then that's what we basically call as the bioprecipitation cycle. Then what next is that, of course, then they do affect the atmosphere and climate, and then, of course, the human, animal, and plant disease. And that's how they affect the biosphere and human health. Together, we call something like as an ecosystem health. So just like any other aerosol, they do play an important role in climate and in the ecosystem health, but the processes through which they do, it's slightly different and maybe a little more complicated. That's the reason what do we say is that why they are different from other aerosols, right? Because the processes and mechanism or the underlying processes and mechanism are slightly different than the way they affect the different process. And that's what we will see. Let's try to look into the sources, right? Until now, you might have seen that the aerosols, when they are coming, like power plant, vehicle, plumes, and then whatnot, like, big plumes coming, like the pollution and everything. Now, interestingly, what do you see? The basic contrast between probably what you might have studied or looked into over the last 4 days, that what are the different sources of aerosol. None of that source really appears here, right? Isn't it? Is it true? Right? Because you don't really have any biomass burning, vehicular emission, factory, and then so on and so forth. And these are primarily the sources of the bioaerosol. And then this is something which is really the man-made. That's the reason I say that it is really an anthropogenic source. And then, of course, you have this biota, and then, like, the bubble bursting mechanism from the waves, where you get this bacteria which are kind of thriving there in the sea, so on and so forth. So these are just the kind of different sources where then you can attribute it that which one you want to call it as a natural source and which one you want to really look into this fact that it is the anthropogenic source. All right. How do you basically then characterize, right? You know, there is a interesting world, a very systematic, well organized, and then very surprising world which really goes down there when you really go below 20 microns, right? You know, and this is what some of the pictures which I want to show you, that these are pollens, right? You know, sometimes when you have this thing going on, and then if somebody is kind of having that grass mowing machine and then doing something with the grass, you get that very typical smell. That's the grass pollens which you have around, right? This is something the protozoa, this is the fungal spores, like, you know, how nicely it is organized. These are the bacteria, different, this is algae, how it looks like. And this is the virus which it looks like, right? You know, so there is a very well organized world there, and that's how they play an important role. Let's say, for example, like, you know, cloud formation, which I think Professor Shweta will talk a little bit more about how the biological aerosols, they initiate the cloud formation through the ice nucleation process. So I'll leave it to her to talk about this. Anyhow, but then what it says, characteristics, whole biological units, then you have something called the viable and then something which is called as the non-viable. I don't want to get into the detail of a microbiology class here because we are at a meteorology institute. And then, of course, you have the fragment, then you have detritus, and then you have, of course, excrement. And all these factors basically contribute, like, you know, animal gender, plant gender, that itself would call as the bioaerosol. Of course, how do you define the diameter for these aerosols is totally different than how you define the diameter for the other aerosols. All right, then why study the bioaerosols? Of course, the very well-known figure for everybody, I believe that many of you might have seen this figure before. Like, you get the largest uncertainty in the current and future understanding of climate change because of the aerosol cloud uncertainty. Why? Because the constituents which warm the Earth's surface, their error bar is much smaller than the estimate itself. But when it comes to the cloud adjustment due to the aerosol, that this error bar is almost double the estimate itself. So when you try to calculate the total effect of this error bar translates as it is. And then that's why we call that the aerosol cloud interaction represents the largest uncertainty in the current and future understanding of climate change. Now let's look at this figure, very familiar figure, that if you have a pristine environment, you have very few aerosol for a given liquid water content. So the cloud formation starts. And once the cloud formation starts, the collision and coalition's efficiency is kind of low in this case. And then so the particles, they grow big enough. And then even before your cloud crosses the sub-zero temperature, the precipitation occurs and the new cloud formation starts. However, in the polluted environment, you have, for the same liquid water content, you have more aerosols. So your collision and coalition's efficiency decreases. So the cloud droplet there are numerous cloud droplets, but they small in size. So they cannot initiate the precipitation. So the cloud really then crosses the sub-zero temperature. Once it crosses the sub-zero temperature, the ice crystal they form. Once the ice crystal forms, it releases that latent heat of freezing that adds more energy into the cloud, that we call it an invigoration of convection. The cloud goes up further and then you get more water. And once the situation or the condition becomes optimum, it just kind of opens the tab, and that's what you basically call it as the extreme event or the cloudburst something. Now, what happens here, if you look into this particular phase, this can take something around up to 3 hours or so. Why? Because unless it crosses the sub-zero at a certain temperature, the ice crystal cannot form. But believe me, that if you have the bioaerosols here, it can initiate immediately at around temperatures of -4, because we know that there are certain bacteria, which probably she will show, that Pseudomonas syringae, they can initiate the ice nuclei formation at a temperatures of around -5 degrees centigrade. So this 3 hours could essentially happen in maybe 20 minutes or so once it crosses the sub-zero temperature. So what it basically means that you are preponing the onset of your precipitation. And that's why sometimes the climate models, when you want to deal with the precipitation, you need to understand the parameterization related with the bioaerosol which are currently very crudely represented into the climate models. Anyhow, so how does it basically work? I'll try to set some stage for Dr. Shweta's talk, like she'll probably do it more details. Then how do these bioaerosols then initiate or prepone this precipitation? Well, thermodynamically, it's slightly difficult, but if I were to just tell you very easily that if you look into the supercooled water, right, the water which is at less than 0 degrees centigrade, it can remain water at sub-zero temperature if your water is clean without any impurity, because you don't really want to provide any surface so that the freezing process can start. Now, what happens is that if you look into the supercooled water at T is equal to 0, then after 48 nanoseconds here, that the nucleation, it starts occurring. Now, if you have the nucleation, like any sort of a disturbance, which kind of start organizing the molecules in that way, and then immediately within no time, within 500 nanoseconds, you obviously can have the structure, which is kind of becoming the ice crystal. Now, of course, you need to have the change in the latent heat there, then only these kind of structure can form. Now, what bioaerosols they do is that they are floating, or the surface structure is in such a way that particular bioaerosol with that protein is very efficient at removing the latent heat from the water, right? Because what is the phase change? If you look into the Gibbs free energy diagrams into this delta G, when the phase change occurs, is that, of course, when you move from your left-hand side to the right-hand side of the curve, obviously you are getting the phase change. And that's what exactly it happens here with the bioaerosol. Now look at this. This is the temperature in degrees centigrade. And then this is the number of ice crystal you can save a centimeter square. Now for the non-bioaerosol, you need the temperatures of around like minus 35 to 40 degrees centigrade to have the heterogeneous nucleation of the ice crystal to happen. However, in terms of the bioaerosol, look at these numbers. It can reach at 10 to the power 6, even at a temperature which is less than minus 5 degrees centigrade. Whereas for the bioaerosol, to get non-bioaerosol, to get that number, sometimes you may have to go to the temperature, which is at around minus 35 degrees centigrade. So let's try to understand that these guys have some sort of a magic wand with them, which basically initiate the ice crystal formation much faster and earlier than the non-bioaerosol. And that's the reason these are the unknown processes which we need to then look into the cloud formation, that how can this happen at a temperature which is like minus 3 to minus 8 degrees centigrade. That's the climate impact of the bioaerosols. Health-wise, of course, they are more important not only for you, but even for the plants. This is a very nice paper. I highly recommend this paper, Fisher et al. In 2010. It's a Nature paper. They have just a very simple analysis, like, animal infected fungi and plant infected fungi, how they are basically on the rise, right? So infectious disease due to fungal attacks are on rise, basically, right? That's what they call for cry for attention, because fungal spores are, unlike the bacteria or viruses, they don't need any host to survive. So any specific fungi which is sitting here and then can travel like 2,000 kilometers, and then if it is not damaged environmentally, can go sit and then can cause the damage at that place, right? So if you don't really care about the fungal attacks on the plant, maybe 25 years down the line, we would be talking about the plant pandemics than the human pandemic. Of course, then this is what something which we have tried to look into over the Indian region, that like how the increasing crop attacks are happening. That's the paper with this is the Yadav et al. Paper. Like, how do you basically see that different type of fungi is affecting the different type of crops which is there. And this is done from the publicly available data. I forgot the name. What is it called? Promid, yeah. So this is done from the publicly available data. There is a very nice mailing list where they give you the information about the alerts about the different type of attacks. It's called the Promid database. Anyhow, so this basically affects the thing. And then I'm not talking about the human health here, because that's a very complicated thing. The only thing which you also need to understand, that the allergies or any sort of a health effect which happens due to the bioaerosol is slightly different than the normal aerosol, because for the normal aerosol, you need to have some sort of a threshold limit. You say that, oh, if it is more than X microgram per meter cube, then only you will have the health effect. Then that is more related with the urban region, right? Rural region is relatively clean. But unfortunately, bioaerosol doesn't know that boundaries, right? If it has to cause an allergy, even one fungal spore you inhale, it will kind of ignite or it will kind of have some sort of a process in your body that will cause the allergies. Whether you are inhaling it in Pune or whether you are inhaling it in Mahabaleshwar, it's going to have the same impact. So unfortunately, like your other aerosols, as far as the health impact is concerned, the bioaerosol does not know any boundary and they can cause you a problem anywhere, even in your indoor. Like, sometimes when you go to some places, you see some sort of a black spots in the bathrooms, right? They are molds and they are part of the bioaerosols, right? So be careful about that. Let's try to get to the main business now about the measurement techniques, the offline techniques. And then I said that it is advantages and disadvantages because personally, I believe that the offline techniques have more advantages than the disadvantages. And in online techniques, they have more disadvantages as compared to the advantages. That's the reason I shuffled these two words here, that advantages and disadvantages and disadvantages and advantages. Let's go over to the offline technique. You can use various offline techniques, the chemical, of course, the physical, the biological, and then the microscopic. Generally, microscopic, I used to talk in terms of the physical characterization. But then when I discussed in my group, they said that, well, okay, you try to separate it because you could get more information than simply the physics out of it. That's the reason I called, because it's a combination of physics as well as the chemistry. That's the reason I think the microscopic is different. So just run through over it differently what happens. I found this very interesting paper a few days back. It was probably I really like the way they have presented it, that how the biosampling and collection techniques can happen. You have impaction, impingement, filtration, gravity sampling, electrostatic precipitation, cyclone separation, thermal precipitation, and condensation. And then, of course, transformation to analyze and categorization. And then how do you basically do that? So you have the culturable approach and non-culturable approach. And then this is where sometimes I do not really like, why do you really want to classify them? But then that's fine. A lot of people, they tend to work as far as the bioaerosol is concerned, simply to the one which you can culture and then the one which you cannot culture. And then, of course, you have this list of different techniques which you can use and then try to, interpret the results. With the bioaerosols, unfortunately, it is not the analysis or the technique which we use to get the data. That's not the difficult part. The difficult part is once you get the data, what to do with it and how to interpret that. What do you mean by that data? That's probably slightly more complicated than simply looking into this fact that how to do that. This is a classic Bauer et al. 2008 paper. That's really one of the first paper where they try to do the chemical analysis of the fungal spore. And then they simply correlated the number of spore with the arabitol and mannitol, right? And when they said that, it's basically come from the bioaerosol. Why? Simply because of the fact that at least until then, I don't know at this stage what the status is. So I'm sorry about it. No other sources of these compounds were reported. So it was presumed that arabitol and mannitol would only come from the fungal spores and not from any other sources. So then you simply go to the FID, GCMS or something, some technique, have your known samples, like your whatever that reference you call it, and then if you get an arabitol and mannitol, try to correlate it with fungal spore. This is a very nice correlation which they have found. So this is now how you do the chemical analysis. Just a typical chemical analysis. Do it. Whatever the tracers which you found, if they are associated with any sort of a biomaterial, you could basically attribute them to the bioaerosol. But before that, of course, you need to collect the samples, right? So then there are different ways. And as I said, that there is no as such standard way that how do you basically collect the samples. You have different things to do that. You have cyclone sampler, you have Schilling aerosol collector, you have then you kind of put it into this 3-piece filter cascade. You have AGI 30, again, they call it an impinger, right? That glass, this thing which they make it is called an impinger. Then you have filter, this slit sampler, of course, that is also very important. And then you have this typical something called as an Anderson sampler. But it's a sampling technique which you also use it for normal aerosol, right? But then what it makes it different is that how do you basically use it for your qualitative and quantitative analysis, right? So I just removed it here and then added this thing. So it requires the post-sampling qualitative and quantitative analysis of bioaerosol. So your sampling in the beginning may not be different, but then once you sample it, it totally changes the course and gets into the different direction altogether. You can use the morphology using microscopy, particularly the electron microscopy, and I'll show you some of the results associated with that, like very interesting stories I'll tell you out of it. The most important thing which we do, particularly we are very proud of, because we have been successful in developing a protocol where you could actually extract the DNA directly from the filter sample, and then you don't really need to grow them, right? So that's something which I would talk about. Culturing using specific substrates, like if you want to kind of target certain specific fungi or bacteria, whatever it is. Quantitative analysis, culturing and count, that's the oldest one. Like you grow them, the colonies, and then take some that scale. What is it called? I don't know. You put it that, and then call the colony forming units. That is what you call it as a CFUs. Then a hemocytometer, of course, that's what it basically measures. It's a simple scale, and then you put it on that, and then it tells you how many colony forming units essentially you have. Fluorescence microscopy, that's what I'll talk a little bit about. And then fluorescence spectroscopy, that's also I'm going to talk about a little bit. So let's start with the microscopy to better understand the morphology using the SEM. One of my first students, she did this study, which is published in Atmospheric Environment, a 2014 or 2013 paper maybe. So she basically came up with a very interesting idea. She used the syringe filter holder. So these syringe filters are basically used by the medical people to kind of do some sort of a filtration. So we wanted to have a very specific way of sampling it. So she came up with this syringe filter holders and then used the nucleopore filter for it, right? Which was probably used by medical people for some sort of filtration. And what she did is that she did a very simple, the simplest form of sampling. So she put a filter holder of 5 micrometer here, and then another one was 0.2 micrometer here. So the bigger particles were trapped here, and then the smaller one came here. Then she took that nucleopore filter and then just looked into under the ACM. And this is what basically she found a very interesting, the particle. Then the point was, of course, that how do I identify? I mean, if tomorrow I say this and then say that this is the, if I don't tell you that this is the bioaerosol and tell that, oh, this is one of the holes which was found on Mars, I can still sell that, right? No, you say that like this is one of the part on the moon, I can still sell that, right? No? So the problem comes in, how do you identify it? Well, that's the beauty of the climate science in general, or particularly the bioaerosol, that you have immense opportunity to collaborate. Always remember that more you share, more benefit you get, right? That's you all are young people here. Never say that it is my data, it is my instrument, it is my this thing, right? More you share, of course, more benefit you get. So what we did is that we looked into all these aerosols, these bioaerosols, that we had no idea what it is. While searching for a lot of literature and people around, we found one very old man in Australia who has been collecting the images of these different bioaerosols for a very long time in life. And then when we sent this picture, he said, wow, this is amazing, right? This is what I was looking for. And then he really classified us that what kind of pollen it could be, what kind of bacteria we did not see, or what kind of spores it would be. And then interestingly, what we found is that not only that it helped us to identify it, but that person really even helped us to see that what is the composition at two contrasting locations that this bioaerosol could be. Just that Chennai and Munnar, one is urban and one is pristine rural. And then, of course, that is really highly acknowledged paper that by simply using the microscopy, we could really get something like this. Microscopy, that is what I talked about, is the scanning electron microscopy, but you can simply use the supplement it with the bright field microscope, the simple microscope. But that does not really give you some sort of a scientific interpretation which you want to draw, but it could still give you a very interesting result. As far as the fluorescence microscopy is concerned, I'll talk about it. We have one in our lab, but then I'll try to, this is what this hemocytometer really looks like. You could actually count how many colonies are there like this. Let's try to talk a little bit about the microscopy and then now the fluorescence microscope. Any biological material, by virtue of presence of NADPH or NADP2, which gets converted into NADPH, if you kind of incident some light on it, of course, the electron excitation happens, right? And then what it basically does is that, like chlorophylls, for example, photosynthesis, why the plants, they basically look green? It's because of the NADPS, the chlorophyll, which they do the photosynthesis. Then some of the energy which is used to do the photosynthesis, but some of that can be used it goes as a fluorescence, right? So there is I think around 700 nanometer or around that range, they kind of emit it. And that's what happens when you put a light on the fluorospore. It basically excites and then does this fluorescence, the quote unquote fluorescence. So what it does is that we have the fluorescence microscope. This is a very classic paper in AMP by Christopher Fielker at Max Planck Institute. I know that he did a very extensive study about it. And then what it does is that you have an excitation, that you excite that fluorospore of a specific wavelength and then try to map the emission, right? And this is how you get a very simple kind of an excitation emission map. And then what it really does is that it specifically you could try to categorize or primarily try to identify whether the bioaerosol emission excitation map looks like. And then if you do some sort of a calibration, like, for the known type of a biological aerosol particles, you could use this emission excitation map really to at least identify whether what you are observing is a fungal spore or whether it's a pollen grain or whether it's a bacteria, so on and so forth. So what they did is that this is how the online technique is developed. Let's try to spend a couple of, I still have 10 minutes, right? Let's try to spend a couple of minutes on this particular figure. So this is the emission and this is the excitation. These are the different online instruments. I mean, let us try to focus on the UV APS. This product is now stopped, not available anymore. WIPs definitely have. So what it basically does, that if I look into this black line here, so the UV APS has some sort of an excitation wavelength at about 370 nanometer, I believe. And then the emission basically starts from 420 to up to 580 or so something. But then what happens is that since you have a different channels for the emissions there, it gives you the emission only in those different wavelengths. And you can't really separate that whether then it is a fungal spore, whether it is a bacteria, or whether it is a pollen grain. And that's the disadvantage with it, because it gives you an emission in this particular range as a continuous function. You can't really say that, although they have a different channel, 64 channels in that. So the intensity may vary slightly depending upon the type of the bioaerosol at a different wavelength, but it's still a very crude way of doing. And that's the reason online measurements can't really tell you that whether it is a bacteria, whether it is a fungal spore, or whether it is a pollen grain. And that's the disadvantage. This is a new instrument called WIPS. I think it has 2 wavelengths, I believe. And then that's the reason it gives you the emissions into, here is the one. It excites it at around 380 nanometer, and then you have like 400 to 600. And there is another one which is at 280, which gives you from 320 till 580, something like this, right? So that you can differentiate. But it still really doesn't work. And ask me after the lecture that, like, what are the different disadvantages as far as this instrument is concerned. Why did this happen? Yeah, okay. So bioaerosol sampling now, it is with one of my favorite things, that DNA analysis. And I quickly run through this. You collect the biomass on a filter paper, then cultivation and DNA extraction. You can directly extract the DNA from it using the DNA extraction kit, and that's a pretty expensive business. Then the quantification of gDNA, and then you use the nanodrop spectrophotometer for whatever that technique is. Then you have the PCR amplification, that you get a very small quantity of the DNA, so you need to have the polymerase chain reaction to amplify them. Then you kind of put into thermocycler. Either you can go for the next generation sequencing. Then you, of course, do the library amplification for it, quantity and quality check. Or you can then go into the, this flow cell to even better understand the importance and then the quantity of it. Or you do the standard Sanger sequencing, because the difference between these two is that you can quantify your DNA in as far as the next generation sequencing is concerned. Then you go into this blast thing, so that if your overlap is more than this percentage, then you can obviously say that the species level identification which I'm doing is correct or not. So one of the ideas to do the DNA analysis is that you can really go up to the species level identification, meaning that what kind of a species of a bacteria or the pollen is or the fungal spore is flying in the air, that is possible. But this, I mean, it's slightly tricky. It's not really that one lecture or 20 minutes I can try to tell you. It probably requires a full workshop again on the bioaerosol characterization, not only one lecture, but the entire this thing. And then you need some sort of anybody microbiologist here? Congratulations. I think she deserves an applause for it. She's kind of coming with this. Anyhow, so this is very easy. So contact her after the lecture, not me then, right? So she can try to explain you. Again, this is the methodology which I'm kind of just trying to go again and again through over it. This is just an example which we did the study in I don't know if this was Jammu or Gurdaspur, right? That was done with in collaboration with Dr. Shweta. It just has a pictorial representation of what other things which you could do as far as the DNA analysis is concerned. Total DNA, NGS, advantageous and sensitive, Sanger sequencing, laborious and not as accurate as NGS. But then this required more effort. Like you truly need a microbiologist to do that. I'm a physicist, so I can tell that. Yeah, this is the DNA extraction process. I wanted to go more details about it, like how do you do that, and then you have the PCR amplification. Then you obviously have cloning transformation and selection, and then you go into the blast, put your sequence there, and then you have to do the screening also. Once you put that into the blast, then you identify it, and then you have to calculate the OTUs, which is operational taxonomical units, to basically quantify your DNA, so on and so forth. Anyway, this is how it basically then looks like. By the way, the results, they really look interesting. What I know here up to species level now, that how much percentage of my ascomycota is there, what percentage of basidiomycota is there. This information is really important. This is for the fungal spores. And then you can, and these people, they make very nice and interesting heat maps, which I need some more time to understand and digest what it basically means. But these are our results from, I don't know, Shweta, do you remember if these are from Gurdaspur or Jammu? Gurdaspur, yeah. Anyway, so then you can, what I'm trying to show is that, I mean, it's complicated. I'm trying to scare you here, right? It's very scary. That's what exactly I'm trying to tell. My whole idea of these slides is to scare you, right? And that's what it really looks very complicated. But then this is what I'm trying to say, that the DNA analysis gives you much more information, which is really difficult to interpret, and then you need a whole bunch of collaboration to interpret this, right? Anyway, so this is what we have done, that different OTUs with this. Yeah, this is how it really looks like. Different diversity than expected. All right, I'll spend another 5 minutes to close with the online technique, because I said that they have more disadvantages than the advantages. So this is a classic instrument which was used widely by a lot of people to look into the number size distribution of bioaerosol. This instrument is called as an UV-APS, ultraviolet. So it's very simple technique, like you accelerate a particle through nozzle. Then you have two diode laser beams. So you know this distance. So how much distance, how much time it takes to cross these two red lasers, depending upon time of flight, you can know the size of that particle. And then once they basically expose it to UV laser beam. Now, once you expose it to the UV laser beam, if the particle is of a biological in nature, then it will give you the fluorescence. And this is how they constructed the number size distribution of biological aerosol. And this is how it looks like. You have the date, you have the diameter, and then you have the concentration. So this is basically the fluorescence. So based on these techniques, right, at what wavelength, or depending upon the size, because you don't have the wavelength dependency here. So then we try to say that, okay, nominally there is something which is at 3 micron, could be essentially the fungal spores. Anything which is around 1 micron could be bacteria. If we find anything around like 15 micron, could be pollen. But that's the reason there is a question mark in that. That's where the online instruments, they fail to tell you that, if you can categorize them, even the WIBS also is not really able to do it. Anyway, so this is how this data again looks like. I'll not go into the, so UV-APS has an excitation at 355, and then the emission is between 420 to 575. But although it has different channel, you cannot really look into the different intensity to try to find out that what kind of that particle would be. This is how it looks like. Some examples, like, you could only look into the size distribution here and say that, okay, this peak probably belongs to bacteria or small pores. This peak probably belongs to the fungal spores. That is confirmed. This is pollen grain in this side. And then you could simply quantify the total number of and then the fraction of bioaerosol. What I see is that, yes, of course, Amazon being the the forest, the tropical forest, of course, will have a higher fraction of bioaerosol. And then nothing more can really be looked into that. This is how the simple results they look like. Anyhow, now just to wrap it up, how to find a suitable technique? That's the question which everybody has. And then the answer to that is, I don't know, right? Because it depends on your objective, and then it more depends on how you are going to interpret your results. Like, so you need to probably have the reverse approaches in that, do your analysis, apply certain techniques, and then try to interpret your results in a proper way. So as far as I am concerned, I do really not know that what kind of an appropriate technique essentially you can apply based on your objective. That's not probably the way it hardly works. Well, just to summarize it, like, bioaerosols, they have a lot of challenges, like, in every aspect of what you do, as far as the health effect is concerned or the climate effect is concerned, you probably need to account them in a totally different way, whether it is an air pollution. So they say that the bioaerosols in an urban environment can enhance the allergic ability of them because of the protein nitration. We can talk about it at a later stage. But then, yes, whether it is a health impact or a climate impact, of course, you need to pay more attention to the role of the bioaerosols in general in understanding the underlying processes. And for that purpose, of course, you need the better bioaerosol characterization techniques. Yeah, of course, thank you for your attention. If any question, I will get.
Any questions? Microbiology.
We can take up few questions. Any questions?
There is always a problem that if you, after a talk, if you don't have any question, there are two possibilities, right? I mean, either you understood everything, which I don't believe, or you didn't understand anything, which is bad for me, right? I will initiate. Mike, you can take the mic so others can.
The bioorganism initiates the heterogeneous nucleations very early temperature, I mean above the 0 degrees Celsius. Why is that? Because for normal aerosol it goes around -35. For bioaerosol it goes beyond -5 degrees Celsius. So why is different from bioaerosol?
But there are two answers to it. One is that she will talk about it in her lecture, that why it happens. But I try to do that. So what happens is that if you look into the thermodynamics of a phase change, like, Gibbs free energy, that standard graph, you remember that, right? So for any phase change to happen, there has to be some change in the energy of the system, the enthalpy, which you call the entropy. Now, if you look into this Gibbs free energy, like this typical curve of the phase change, now when you have a super cooled water, they need some sort of that system has some sort of an enthalpy and entropy, whatever you call it. Now, the bioaerosol, based on its structure, the protein structure, or for example, the shape or the surface properties, when they get in touch with these supercooled water molecules, right, they are very effective in removing that latent heat. So once they absorb that latent heat, I mean, and we can talk a little bit, there is a very nice paper by the Maxman group that why it basically happens. So once they remove that latent heat, of course, then your system, the thermodynamical property of that system changes, and that initiates the phase change, right? So they are more effective in, I mean, I can only tell you that due to the constraint of time, that they are very effective in removing that latent heat, and that initiates the phase change. Those other aerosols which you are talking about are not that effective in removing that latent heat.
Basically, surface property of the
Yes, exactly. Both surface property and the composition, that protein and other.
I have more of a general question. Like everybody is talking about this health effect of aerosol.
Here.
Loud.
So everybody is talking about the health effects of aerosols. Okay. Now I'm seeing like health effects of bioaerosols. So when we compare like bioaerosols versus atmospheric aerosols and seeing the health effects which we are studying right now, like cardiovascular diseases, like cardiovascular disease, like cardiovascular and like you can say mental health, and all these are related with the air pollution. Isn't it relevant to say that these are like of not very significant proportion, the effect of this air pollution, rather than other factors are there that are having more effect, like comparing this atmospheric aerosol to health effects?
Well, I mean, yes, I mean, of course, the sorry,
Other factors are more dominating than these.
I mean, the answer is yes and no. I mean, of course, the bioaerosols are also atmospheric aerosols, right? But then when you come to a very specific environment, right? Let's say indoor environment, I would say that it is very important to look into the bioaerosol, right? You know, in molds, for example, at home. Nobody really talks about it. Particularly I mean, don't quote me outside. I don't want to get into media for wrong reasons. But the airports,
It's getting recorded.
yeah, it's getting recorded, but not that it is getting this thing. Let's say, for example, airports, right? Or any particular place which has no proper ventilation, right? You know, this is where the very large debate came about the COVID-related thing. That the first thing is mask, period, right? You know, the mask really reduces, and second is ventilation. And this is very basic. Like, you know, if you go to places like airport, right? You know, I don't know if they have a good ventilation per se. So I would really be interested if the permission is given me to kind of sample within the airport and see what all is in there, right? So again, that your question depends in what perspective you want to see as far as the health effect is concerned. I don't think there is any standard answer to that, saying that ये classification नहीं होना चाहिए, ये होना चाहिए. I don't think so. Hold it close.
Hello. It's a very basic question. So, are these bioaerosols acting as a CCA, or are we just absorbing the energy and giving rise to the situation?
What is the question? I can't hear. I have 30% less. Yes.
Are these bioaerosols acting as CCL or
Acting as
CCL,
CCL, yeah.
or they're just absorbing the energy and then giving rise to the situation?
So they do act as the giant CCL.
Okay.
Because their size is really very high. Like, you know, so some of these bioaerosols, of course, through the transformation and aging, sorry, I missed that point about the giant CCL, right? So they do act as a CCL, but they may not be in the pure form of the bioaerosols, really. So there are these giant CCLs, which are mostly dominated by the bioaerosols.
So,
They do.
so the condensation is happening on the
Yes, exactly. They provide the surface in the sense that, of course, the condensation happens.
So is there any technique by which we can measure the viscosity of such aerosols?
There is a very interesting paper by Mikhailo et al. In AACP. I think it is a 2022 or 23 paper, right? You know, where he really looked into the water uptake by pollens. I can give you that paper. You could do that. There are multiple ways to do that, but you should really refer to that paper by Mikhailo et al. I'll give you that paper.
So the hygroscopicity will vary.
Yes, exactly. It does.
Whatever chemical composition.
Correct. I think he has done it for the pollens. I don't recall, but for the pollens, yeah, there's a very interesting paper. You should read that. Correct.
Thank you.
Okay. Thank you, sir, for this nice lecture. Sir, in the graph that, in the heat map you were talking, that it is scary. So in what terms do you mean that it is scary? Like in terms of statistical type, because we have to classify each, we need a huge library first, and we need to classify each bioaerosol DNA sequencing, or it means we need a huge collaboration and everybody might not be exposed. Like what is the scary part?
Yeah, well, okay. The scary part is that because I'm not a microbiologist, I'm a physicist, right? So maybe then she's the one who has helped us more about how to prepare those plots and then how to do the. See, the point is that the information is so overwhelming there, right? You know, you have to be very accurate about how do you interpret those results. And then I would be very scary that if I get overwhelmed with the information which I don't know. So I think as my student has worked with Dr. Shweta to really fine tune how do you represent it. That's the reason I say that it is scary for me, because you need a really very, you don't want any wrong information to go to the literature. That's the reason I said that it is more scary, because I'm just a physicist. That's it.
And so, when we are talking about sampling, like we have the classical cascades and impactors. So and there is other scanning that like sort of first time UV APS. So can we just couple it for a like near real time kind of sampling that we know actually this is the silent. Then we have sampling.
Yeah. People, they are doing it. There are multiple ways of doing it. You can do all the permutations and combinations, no problem. You sure?
In 2001, there was red rain came, right? Red. So some people some people say it was because of RT. I would like to know your opinion.
No, I mean, I think, yeah, I have heard about it. We're trying to do something like, it's probably some sort of a circulation of some specific material, right? I don't think there is anything scary with that, but I have no idea, honestly speaking, right? Unless I take that rainwater and do some analysis, I think it would be wrong on my part to comment about it. But I think, yes, I mean, some sort of that biological material probably it is in there. But it's very wrong for me to comment it without knowing it. But yes, I'm aware of something which is called as a red. If it happens next time, I can promise you that I will get the samples and do the analysis.
Like compared to other foreign countries,
Other?
foreign countries, foreign countries,
Foreign.
Okay.
So, do India have that much kind of pollen? Like, I think pollen allergies are more common in foreign countries. Right?
Well, I mean, see, that's a different, again, the discussion. See, the allergy is concerned that every human, it kind of responds to the differently for the allergies, right? But then, yes, the allergies are anywhere same, essentially. It's just that maybe we don't pay that much attention, right? We are not so much conscious towards the allergies. That's the reason we say that maybe we have it less. It's not the case, right? We need to pay more attention. And then I kind of got involved into the allergies because my daughter once got and she was in ICU for 1 week because of the allergies. During the COVID time, we started knowing that the oxygen and this thing, at that time, her oxygen has gone to 79 because of this flooding of the, this fluid in the lungs. So we need to, I mean, it's good that you brought it. We need to become more and more conscious about allergies. That's why we do have it. It's not that it doesn't register in our mind. We are not conscious about it.
Thank you.
For any further doubts, you can discuss during the tea time.
Exactly.
Thank you, sir, for creating our knowledge on bioresources. Our second lecture on INP sampling and characterization will be delivered by Dr. Shweta Yadav, a faculty in the Department of Environmental Science, Central University of Jammu and Kashmir. She has PhD from JNU, New Delhi, and postdoctorate from NC State University, USA. She was awarded the Fulbright Climate Fellowship for postdoctoral research in 2017, and she has got selected as member of Indian National Young Academy of Science INSA for the period of 2023 to 28. She is also working as technical partner for Jammu and Kashmir Pollution Control Committee under National Clean Air Program (NCAP). Her research group is developing understanding the chemical, biological, cloud forming, and ice nucleating properties of aerosols to understand their implications on the Himalayan climate. With this, I invite Dr. Shweta Yadav on stage to deliver her lecture.
Good morning, everybody. Am I audible? Good morning. Good morning. Good morning. So last lecture of the workshop. At the outset, I'm very thankful to the organizers, Dr. Panditharai, Dr. Padma, Dr. Manish, IITM for having me here. I'm very grateful to the organizers to invite me because this is my first visit to not only to IITM Pune, but to Pune. And I'm very happy to see, in addition to my collaborators, I'm very happy to see some faces which we have met in other places, like Simran is sitting there. She was part of our winter school in Jammu, and other students also. So it's a very happy feeling for me because see, when we are at this side of the platform, we generally either we talk in a conference, we are sharing our research, we are in some meeting. But when we're in front of some students, it's a it's a ray of hope because see. It gives you a happy feeling that whatever I have done, I can share with you, and whatever you are doing, it can help you. You can plan your future research, and in somehow, some way, you are going to use whatever we are going to discuss. So with this, I have two news to share. One good news is that my presentation is very short, and the bad news is that you have to be with me. So you are going to be with me?
Yes.
Thank you. So I'm going to talk on sampling and characterization of ice-circulating particles. I'm leading aerosol research group in Central University of Jammu. So our students are also participating here in this winter school. Oh, sorry. Winter school is so much on my tongue that anyways, the training workshop, I call it winter school. So yeah. So I think you already know the basics of nucleation. Dr. Panditharai is here. So you have already seen spin during the Mahabaleshwar visit, and you have all the basics. And I was, when I was preparing my slides last night, I was looking at the YouTube videos of this training workshop, and I saw that these are really exhaustive, and you have understood very, very basic things, I hope, about atmospheric aerosols and the different aspects. So one good thing of isolating particles or bioaerosol is that whatever you are doing on aerosol, and if you want to expand your horizon, you can use these parameters for understanding the processes. So during my talk, I'm going to look back a bit about history of INP, fundamentals of isolating particles, strategies for measuring INP, principles of INP measurements, and available instruments for INP measurements all over the world. So yes, ice-circulating particles are a subset of atmospheric aerosols that can initiate primary ice production and trigger a cascade of microphysical processes. So we say atmospheric aerosols are omnipresent, and I say INP are omnipresent. And he will say bioaerosols are omnipresent. So all are omnipresent and all are complex entities. Looking back at the history of INP, this is a very famous paper by Kredenweiss. By the way, Marcus Peters is my mentor, and Sonia Kredenweiss is Marcus Peters' mentor. So this paper is very good, and this paper is so comprehensive that I think if you start reading it today, you will spend your entire PhD reading it. And so many concepts it will give you. So I recommend that you should go through this paper. So see, basically, I am an aerosol chemist. So I am a chemist, and Professor Sachin is physicist, and we are doing bioaerosol ice-circulating particles. So this answers your question of collaboration and interdisciplinary research to understand the processes, right? So I started my work on INP way back in 2016 when I got this Fulbright fellowship and I got a chance to work with Marcus Peters. Till then, I was focusing on chemical characteristics only. And you will be surprised to know that I was not knowing about ice-circulating particles in that detail. I was knowing about cloud condensation nuclei, a more common terminology. But when I was reading this paper, I got to know that the knowledge about ice circulation started in way back in 1940s, right? So initially we were calling them sublimation nuclei. Then way back in 1940s, we got to know that these nuclei are insoluble, wetted with water, and susceptible to deactivation by heat or freeze or cycling. So can you believe in 1940 we knew this, and still we say that we have limited knowledge on ice circulating particles? Schaffer 1946 used the term ice nuclei, and in 1947 it was hypothesized that effective ice circulating substances would have crystalline structure similar to ice and identified silver iodide particles as INP. So I believe you must be
a little back. We will see in the camera also.
Oh, I'll visit camera also. Am I looking
It's okay. Yeah. On YouTube it will come so.
Okay. Fine. So, but it is very close to me. It's okay. So, so silver iodide, I believe you must be knowing. It is used for artificial rains, cloud seeding. So basically, in 1947, we got to know about silver iodide. And initially, whatever interest we were having on ice nucleation was just to have artificial rains or cloud seeding and all of those. But I think we were not aware of the microphysical properties, how INP is affecting the Earth's radiation budget, how it is affecting the aerosol cloud interaction. So most of the focus was on the cloud seeding only. And fundamental research by these gentlemen in 1910s to 40s laid foundation of our current knowledge on CCN and INP. And this is a graph again from Kardanice et al. 2019. So you can see here that in 1960s, this blue thing is for ice nucleation, the publications on ice nucleation, red is cloud condensation nuclei, and then black is aerosol in climate. So it's surprising for even me that way back in 1960s, we were having more papers on ice nucleation, ice nucleating particles. Still, we know more about CCN than INP. Then what is the paradox? What is the puzzle behind it? So basically, the initial focus was on cloud seeding, as I shared with you, and lately the significance of INP was realized in cloud microphysics. So when we say CCN versus INP knowledge, the link between particle chemical composition or particle surface properties and ice nucleation are not well established. So if I try to understand the aerosol CCN problem, then I will say, okay, we get a CCN spectra using the CCN instrument. So there is one part of the problem that which aerosol will be behaving as CCN, and other part is how the CCN is correlated to the cloud formation and all of that. Similarly, if I break the ice nucleation problem in two parts, the first part is which particles will be behaving as ice nucleating particle, and second, how true our data is for the cloud microphysical properties. So in the case of ice nucleation, the first part of the problem is itself very complex and we don't have the answers. So this is the difference between the CCN and INP problem. So basically what we are doing, what we can do, we are the people. So I am basically a measurement person. I do a lot of measurements. So we are doing the measurements, and parameterization based on laboratory and field data are therefore used to compare INP concentrations. So the INP spectra, so this is the INP spectra has already been shown by Professor Sachin, but I'll be stressing on INP spectra during my talk. So basically what we get from the INP measurements, we get an INP spectra. So INP spectra can be used to predict a lower bound of ice crystals that is expected at a given temperature in a cloud. So I got to know from the organizers that some of the students sitting here are from masters also. Like, so for you, these things must be very, very new and maybe complicated. But my request is that whatever we are talking here, just try to be with us and try to understand and try to, later on read all these aspects, because when tomorrow you are planning your research, you're designing your PhD problem, these concepts will really help you to explore the new dimensions. Actual ice crystal concentrations inside the cloud may be much larger due to secondary ice processes. So this secondary word is again a very complex word for aerosol scientists. Secondary inorganic aerosol, secondary organic aerosol, secondary ice formation. So basically what is happening because of this secondary thing, everything is again very complicated. So whatever we are getting from spectra, it's fine. But because of secondary processes, what we are getting actual in the cloud is very different. So that gap remains. So what are the limitations of doing all of this INP spectra and all is the inability to represent the time dependence of the nucleation process, the dynamic preferential removal of INPs due to cold rain formation, and the inclusion of a wide range of freezing processes that are not represented in this framework. So what we are doing is we are using several types of instruments to understand the INP concentration, but still these limitations remain with us. So tomorrow, if you're planning your PhD research, you can look for answers for these questions which we have at present. As I shared with you that initially we started with cloud seeding, then we realized the importance of INP in cloud microphysical properties. And recently, if you see that if you are doing any, if you are focusing on any problem in your PhD, so if you're doing chemical characterization or biological, something like that, you must explore the INP potential of your samples. Why? Because the recent highlights and potential sources of INP are very, very interesting. I'm putting here a few. We all know biomass burning is one of the major factors in India for aerosol pollution. So when INPs are emitted in fresh biomass burning aerosols, what happens is that inorganic elements during the combustion process leads to potentially ice active minerals. So what happens is that biomass combustion produces ice active minerals in biomass burning aerosol and water ash. Second thing is, again, the secondary, so once the fresh biomass burning aerosols are emitted from the biomass burning episodes, what happens is there can be evaporation, oxidation, soil condensation, and then aqueous phase, and then the freezing thing. So basically, again, what happens is, this is a new finding. When atmospheric aging enhances the ice nucleation ability of biomass burning aerosol, what happens is it removes the organic carbon particle coatings that conceal the mineral-based ice active sites. So basically what is happening when the aging is happening of the biomass burning emitted aerosols, they become more ice active. So how you can do all of this? You can only do all of this, you can understand all of this when you are having some capability to study INP. You are having some instrument to study the INP characteristics and concentration. The third one, which is very relevant to us as we are in Jammu and Kashmir, the glacial outwash plains are thought to be a major dust source in cold high latitudes, and glacial outwash sediments possess higher ion ability than mineral dust. So these are the different perspectives which makes INP research very, very important. So now I will be talking very fundamental things about ice nucleating particles. Somebody was asking what make bioaerosol a potential INP or something. So when we are saying ice nucleating particles, it is just a specific subset of aerosol, right? And we say 1 in a million particle act as ice nucleating particles. So the curiosity question is, what makes it an INP? What are those properties, right? So the first thing which you should know, which Professor Sachin has already stressed upon, is that INP lowers the energy barrier that should be overcome for the formation of critical ice embryo. Homogeneous nucleation freezing. So I think you already know what is homogeneous and what is heterogeneous nucleation. Do you know or not? So heterogeneous nucleation, when there is any foreign particle present and nucleation is taking place, right? And this foreign particle is our beloved aerosol only, right? And homogeneous is when a pure water is freezing at much, much colder temperatures. So if you focus here, homogeneous nucleation freezing colder temperatures minus 38 degrees C, and heterogeneous, it's warmer than minus 38 degrees C approximately. So this temperature point, some people say minus 38 degrees C, some people say minus 35 degrees Celsius. So don't get confused in that, but approximately at this line, there is a line of mixed phase clouds and ice clouds, right? This temperature delineates the warmer mixed phase clouds from colder cirrus cloud regime. So cirrus clouds, you all know, high clouds, these are high clouds, right? These are ice crystal flaky structures. And mixed phase clouds, this term, I hope you already know what is mixed phase cloud, where we have liquid droplet as well as we have ice crystals in the cloud. So basically, what happens that the MPCs, the mixed phase clouds, are thermodynamically unstable, and most precipitation in clouds initiates through the ice phase. So that's why INP is very, very important. Main characteristics of an INP, to be very, very specific, the particle must be insoluble to provide a solid surface on which the ice germ forms. But whenever I am telling you all these characteristics, these are the broadly accepted. You will find some papers who will show some exceptions, right? Because research is going on and we are developing understanding. But this is the basic characteristic features. INPs tend to be found in the subset of atmospheric aerosol that exist at larger diameters. So generally we say if it is greater than 500 nanometer, it can act as an INP. And the surface should provide sites for hydrogen bonds to form. Ice nucleating surfaces must have a good crystallographic match to ice, and the surface must have active sites as demonstrated above. And surface properties favor lowering of energy barrier to crystallization. So these are the few features or characteristics which make an INP an INP. Bioaerosols as INP has already been covered. Very quickly, I'll tell you that what happens is why we are interested in INP, because it is leading to mixed phase clouds. It is leading to at warm temperatures the freezing takes place, right? So if the foreign particle or INP is not there, the freezing will take place at much colder temperatures, minus 38 degrees C or something. If INP is there, what will happen? Warmer temperature freezing, right? Say minus 15, minus 12, because when I'm talking on bioaerosols, there are other sources also like mineral dust act as INP, different sources, whatever you name it and get it. But why we are emphasizing on bioaerosol? Because if we see the spectra, if we are moving from minus 38 degrees C to towards the warmer temperature, and if you are having a bioaerosol in your sample, then the freezing will take place at minus 4 or minus 5 degrees C. So you can see the shift here. So basically what is happening, you all know lab state, right? Temperature decreases with height. So whatever processes are taking place at very high altitude will take place at a lower elevation. And this actually affects the cloud microphysical properties, hydrological cycle, and all of these. So these are some of the biological ice nucleators. Of course, Pseudomonas, Irvina, all these are the known biological ice nucleators. Other are fungi, algae, pollen, etc. And this is from our recent paper, Kumari and other 2024. In this, we have emphasized on ice nucleating bacteria, ice nucleating fungi, and ice nucleating algae and lichen and pollen. So these are the species which are actually acting as ice nucleating. So basically, there are two ways of doing it. One is, okay, I know now these are ice nucleating. So I will analyze my sample and I will see whether these are there or not. Second approach is I will analyze my sample. I will see if they are there or not, and I'll try finding which are other ice nucleating bioaerosols, right? So expanding the research basically. So IP cloud precipitation connection is now better understood. And if you just see this is one example. This is a paper called Cracking the Problem of Ice Nucleation. However, I believe it is not cracked yet. So here you can see the ice nucleation of feldspar. So showing this figure, I just want to convey to you that yes, INP are very important. Here are different scenarios. So you can see few ice nucleating particles are there, and at a higher height, there will be what? There will be a long-lived ice cloud. At if we see stratus or altocumulus level, many small droplets form a cloud with a long lifetime. So basically, if few INPs are there, cloud will be having a long lifetime. And if you see many ice nucleating particles, what will happen? It will grow and it will precipitate. So basically, this figure is to show you that yes, INP is affecting the aerosol cloud interactions and cloud microphysical properties. When we want to understand the measurements, because the topic which has been given to me is sampling and characterization, we must know what are the different modes of nucleation, right? So we already know homogeneous is pure water when it is crystallizing. Heterogeneous is the different modes are there, and the distinction is made on the basis of whether water nucleates ice from the vapor or the supercooled liquid phase. I repeat, there are subcategories of heterogeneous nucleation, and how we get to know it is deposition, it is this, it is that, whether the nucleation is taking place from the vapor or the supercooled liquid phase. So here is the example. The first mode is deposition nucleation. So formation of ice in an ice supersaturated vapor environment. Next is freezing nucleation, formation of ice in a supercooled liquid environment. And we can subdivide the freezing nucleation. There's some issue, I think, here. Okay. So freezing nucleation can be further classified as fast immersion freezing. So when I come forward about the measurements, you will see that we are doing immersion mode measurements of INP. So a particle immersed in the body of supercooled water, we call immersion freezing. Condensation freezing, as the name says, where a particle acts as CCN and immediately induces freezing of the water condensate. And third is contact freezing. So what is contact freezing? As the name suggests, a particle upon contact with a supercooled liquid water phase will lead to heterogeneous nucleation. So these are the different modes of nucleation which we should know, because then only we can understand what INP measurements we are doing. So this is again a summary of different ice nucleation modes. This is another very interesting paper, overview of ice nucleating particles by Kanzi et al. 2017. Zamin Kanzi is our collaborator also, and he has written this comprehensive paper. If you have any questions related to INP, you must go through this paper. And in this figure, I just want to emphasize that, okay, you can see here, these are the different modes. Homogeneous freezing is there, where you can see above colder temperatures than minus 38 degrees C. And in this window, you are having condensation, immersion, evaporation, collision, contact, etc. So measurement principles of INP. So by now we have understood what are INPs, what are the characteristics of aerosols which make them INP, what are the different modes which are used for measurements of INP, and now we are coming to the measurement principles of INP. So when we talk of measurement principles, I don't know why this is happening, but there is some.
It's just that we raised the button for too long.
Okay. So the first thing is the first category of instruments is expansion or mixing cloud chambers. So have you heard of the mixing cloud chambers? So basically these are expensive to build, require significant maintenance and require skilled personnel to operate the instruments. However, I believe that skilled people are required for operating any instrument. But continuous flow diffusion chamber CFDC. The construction of a CFDC like the Colorado State University design requires significant financial resources and technical expertise to build. And the third is the cold stage technique category. So in cold stage, these are relatively simpler and less expensive to build. Well, the limitation of cold stage instruments is that some groups all over the world are having this cold stage instrument. They're doing the measurements, but there is no commercial version available of these cold stage instruments. So these are the three categories under which we can divide the measurement instruments for INP. And the first is, so I want to confess here that I also have not seen all of these instruments which I'm showing here, but I'm just showing because we work on immersion mode freezing and we work on cold stage instrumentation. But why I'm showing to you this is because if tomorrow you are interested in INP and if you're visiting these places, you can just see them and explore what are these techniques, right? So one is continuous flow diffusion chamber. It is an ice activation spectrometer. So whenever you are talking of INP instrument, you should always see that what is the temperature range it can cover, because we are talking of heterogeneous nucleation. So from which temperature to which temperature it is going, what is the RH regime and all. So here you can see the measurement range of CFDC is approximately -10 to -40 degrees C, and liquid-based refrigeration system provides excellent temperature stability and uniformity. And second is aerosol chamber IDA, that is aerosol interactions and dynamics in the atmosphere. You can see how huge this setup is. IDA is a unique experimental facility, again in Germany, for investigating the impact of aerosols on climate, weather, and the environment. And aerosol and cloud experiments are performed in such a big chamber under relevant atmospheric conditions within a wide range of temperatures. So now you can see a temperature range -90 to 50 degrees Celsius, and then pressure and relative humidity. So this is one of the very biggest, setup we have for INP. And of course, the rare ones. Then another is FINE, which is called Portable Ice Nucleation Experiment, based on the design of IDA chamber and mimics cloud formation upon air mass lifting. So it consists of a 10-liter externally cooled vessel, you can see in the picture. And it is a mobile cloud chamber to investigate INPs relevant to ice crystal formation in mixed phase clouds as well as cirrus clouds. Another is freezing assay ice nucleation spectrometer of the Carlson Institute of Technology. And in this, you can see they do immersion freezing mode. So if you remember what is immersion freezing mode is, when the particle is immersed in the liquid droplet, right? So here you can see so basically for INP measurements, the PCR plates are also used. We just put the drops over there. We allow them to freeze and we see how the INP concentration is changing or the freezing is taking place. So in this one, the general working principle of the freezing assay is that multiple pico to microliter scale volumes of water suspended aerosol samples are supported on a substrate which is cooled down with a constant rate until the volumes are frozen. So in layman's language, what we are doing is if I am having a precipitation sample, I will just take that sample, I'll use a pipette and I'll put 1 microliter or 1 picoliter drops, and then I will allow it to freeze. And then I will see how the freezing is taking place and I'll calculate using the Wallis formula, the INP concentration. So basically, INP concentrations as a function of freezing temperature are calculated by detecting the fraction of frozen aliquots independence of the temperature. So then after this, next is cold stage instrument. So I told you three categories. This is the third category of instrumentation, and this is actually what we do. So during my postdoc, I used NC State cold stage instrument with Marcus Pettus, and since then we were trying to make our own portable instrument. And I'm very happy to share that now it is functional in Central University of Jammu. And so basically, cold stage instrument is offline. So I think you are hearing this word offline and online in all the talks. Professor Sachin also said in chemical characterization also must have, I heard Professor Tripathi, Professor Rastogi, Dr. Manish, everybody is talking about offline and online, right? So what is offline? When we are collecting the sample in the field and I'm analyzing in the laboratory. So somebody was asking me, he was asking me yesterday, what is the height of your university? So the altitude of my university does not matter. Why? Because I am doing the offline characterization. So I'm collecting sample from the field and then I'm actually characterizing it in the university using the offline techniques. One more important term you should know is one is offline, another is online technique. One is destructive, another is non-destructive. So you know what is destructive? The sample is gone, right? And non-destructive, the sample is still there. So like you use optical transmissimeter. So with the same filter, you can do the GCMS analysis as I had done in my PhD. So in some techniques, you can just use the same filter later on for another purpose. So basically in offline, we are collecting the sample from the field and we are analyzing in the lab. So it works on the principle of drop freezing assay technique. A bulk liquid sample is subdivided into droplets, which I just shared with you. So how you will subdivide it into droplets? Just using the pipette. These droplets are placed on the surface. Surface is cooled, and as it freezes, we see how it is freezing with respect to temperature. So it is based on the Poisson distribution. The INP concentration of the liquid is then calculated using the method of Poisson. So you should always remember when we are talking of which type of distribution it is a Poisson distribution on which the INP calculations are based. So this is the cold stage instrument. You can see it is a very, very simple instrument, and the details of this instrument can be found in our paper. So this is the paper which is called Open Hardware Community Ice Nucleation Cold Stage for Research and Teaching. So in this paper, you will find very minute details. Believe me, if you want to have your own cold stage, you can make it. Of course, you have to discuss with us a little bit. So this cold stage has been developed in collaboration with Marcus Peters, with people from RS University, and now we are good to go in Central University of Jammu with the first prototype. But this is very useful because now we are good to go for any offline characterization. So during my postdoc, what I was doing is I was collecting sample and then I was going to US for doing the ice nucleation thing. But now we can do in our lab itself. And what is the benefit? We can study snow sample, we can study precipitation sample, we can study aerosol sample. Sky is the limit. So this is one advantage of using these cold stage instruments. So as I have already said, well suited to study deposition, immersion, and contact freezing modes. To carry out process level studies, probing questions such as nucleation rate, active site densities, because ice nucleating particle field or research is evolving. We are having so many fundamental questions. So using the cold stage, we can answer these fundamental questions also. And importantly, measuring the concentration of atmospheric INP concentration, of course. Since it is collected on filters or with impingers and filter samples are relatively easily obtained, we can measure INP concentration from many popular sampling platforms, like if you're collecting a sample over ship, balloon, or you are using an aircraft or ground-based site, you can just bring the sample to our lab and you can get to know what is the INP concentration of that sample. So characterization of ice nucleating particle, I want your attention on this slide because, see, I am telling you that we take the drops of the samples, we allow them to freeze, but what happens over the process is that the two names you have to remember is one is Big 1953, who gave the drop freezing assay technique, and another is Wali, who gave this formula. So you can see this formula here. We get to know the INP concentration by knowing the fraction unfrozen. So what will happen? Suppose if we are allowing all the drops to freeze over the temperature, some will freeze at some temperature, some will not freeze at some temperature, right? So with temperature, we see that how many drops are like fraction is unfrozen, and we divide it with the V drop. V drop is what? Volume of the drop. So if you are using 1 microliter per drop, it will be 1. So the formula is very, very simple. So this is from our experiments also. In the first figure, you can see these are the sample drops at -5 degrees C. So basically you can see these are the frozen ones and these are unfrozen ones. And if you see, this is the eye spectra. So you, at least from my talk, you should know what is an eye spectra, how it looks. Tomorrow, if you see eye spectra, you should be able to understand, right? So whenever we see any graph, what we see the first thing, we see what is there on the x-axis and what we see that what is there on the y-axis. So the first panel you can see, we are having temperature, then -10, -20, -30, and here we are having the fraction frozen, right? So what will be the fraction unfrozen? 1 minus fraction frozen, of course. So basically you can see this is the HPLC water and this is our sample from Dharamshala. And you can see that the freezing is taking place at around -4, -5. So if it is freezing at -4 or -5, what does it show? Bioaerosol, right? I just told you, bioaerosol. So basically, in the second panel, you see we are having temperature at x-axis and we are having INP concentration per liter water. So these are the results for water and for the Dharamshala sample. So these are actually 3 runs and we have binned the data. So we just do the 3 runs just to see the repeatability of the instrument. And this is how you get the results and you get the INP concentration. So when we started working on INP, we saw that very limited data is there. Some papers are from Dr. Padma Kumari's group also, from Kaipix, and some measurements were done in Pathankot and all. But from Himalayan region, we could not find any ground-based data, and we started the precipitation and aerosol samples in JNK and Himachal Pradesh. So this is the Petters and Wright 2015 paper, which shows all the observations. And if you see that we were not having any such observations from India, and these are our results. So I'm showing you these results that, okay, we are using the cold stage instrument, the offline characteristics, whatever. So what results we are getting, what spectra we are getting, and what information we get from there. So you can see over here, these are the results from Dharamshala, Jammu, New Delhi, and Lucknow. So you can see these are the results of INP concentration. And we can see there is not much difference in background and polluted locations. So whenever you are working in India, the first question comes is what is the effect of pollution, right? So our question was also the same. What is the effect of pollution on INP concentration? So we found that there is not much difference in background and polluted locations. Similarly, Chen et al. 2018 in China also showed that urban air in China is not very different than the background location. So basically, it shows that pollution is not affecting the INP concentration. However, if we do a lot of more, investigations, maybe we get some different answers. But at present, this is one of the status which you should know. And this is another thing which you can do with a cold stage instrument. So what we did was we did time resolved INP measurement. So as the storm progresses, suppose rain starts at 9 a.m. in the morning. So then you can see, okay, you can collect sample at 9 a.m., 9:15 a.m., 9:30 a.m., 10 a.m., 10:15 a.m. And then you can plot the INP concentration here and you can see how the INP spectra or concentration changes as the storm progresses. So likewise, we did here also in the Yadav et al. 2019. And we saw that as storm progresses in monsoon season, INPs are getting replenished. And later biological INP are added. So there are different processes which are leading to all these conclusions, right? So again, the complexity remains same. But yes, these measurements help you in understanding the atmospheric processes better. We also try to see the INP analysis of Thar Desert dust. So this was another question because we found that we were not having any knowledge about the Thar Desert dust and its ice nucleation ability. However, we were having data from different deserts all over the world. So we tried doing this also and we found that Thar dust INP activity is reasonably consistent with the Newman et al. Parameterization. So this is one thing which you all should know that I am saying that bioaerosols are very efficient ice nucleators. They lead to freezing at warmer temperatures. So what is the quick way of knowing whether your sample is having bioaerosol or not? So what you can do is that first thing, when you see the spectra, if you are looking at any freezing at -4 or -5 at warmer temperatures, you can do this quick heat treatment of 10 minutes at 100 degrees C. And what you will find is there is a shift of the spectra. So you can see here, These are the samples before heating, and when we give this heat treatment, there is a shift of the spectra. So basically, because the proteinaceous component is there in the cell wall, it is broken, denatures, and that's why this eye spectra is shifting from here to here. So whenever we are doing INP concentration, it is another very important question that what is the composition of this INP, where from it is coming. So at least one question solved, that if we are giving the heat treatment to the samples, we can get to know, we can confirm whether the bioaerosol is present or not. So our group is actually now targeting to understand the chemical, biological, ice circulating and cloud forming properties. In line to it, we are having this Indo-Swiss project. So I remember 2 years back when I used to give talk, I used to say that we are coming up with the cold stage. And I was saying, like, for how long I'll keep saying that we are coming up with the cold stage. But I'm happy now we are having the cold stage. And for last 1 year, I'm saying we are coming up with this campaign. But it is very, very difficult. But I'm very excited about this campaign. It is in collaboration with the ETH Zurich. Zamin Kanzi and Christy Diner are the collaborators from ETH. And we are going to install all these instruments here. So we are going to do INP, CC, size distribution, chemical speciation, and EDN analysis. So Zamin is having experience of developing ice nucleation instruments. So he's going to bring his HINC, horizontal ice nucleation chamber, and drinks. So these are in addition to other instruments, we are going to have this also for 1 year in the Himalayan region, and we are going to have this comprehensive campaign called Ice Crunch. So this is one example. This is the picture from drinks. So drinks is what? It is a droplet ice nuclei counter Zurich, he calls it. Again, important thing is from which range to which range it is going. So it is, I think I missed 0 there, but it is going from 0 degrees C to below -20. And demonstration of droplets freezing wells can be seen here in this picture. Change in light intensity due to attenuation of light from freezing indicates time and temperature of freezing. This is another instrument which is called horizontal ice nucleation chamber. It is HINC, and HINC consists of 2 horizontal plates which can be cooled to temperatures relevant for ice and mixed phase cloud. So this is very important that in HINC we are going to focus on both mixed phase as well as ice clouds. And as the plates are covered with wet filter papers, a supersaturation with respect to water and ice is established through a temperature difference of the upper and lower plates. So basically there are 2 freezing modes can be investigated in HINC. One is deposition freezing and another is condensation freezing. So basically when we are going to have this ice crunch campaign, we are going to have drinks, we are going to have HINC, we are going to have our cold stage. So we are trying to understand the different INP characteristics at different temperatures and different nucleation modes. This is the whole idea of having this campaign. So coming back to our question of offline versus online measurements, Professor Sachin has already told you advantages, disadvantages. I also second him in saying that if I'm doing the offline characterization, suppose I'm having a sample on my filter paper. So what I will do is I will do the chemical analysis also. I'll do the biological analysis also. So our lab is, we are having one small laboratory called Himalayan Aerosol Research Instrumentation Facility. So we can do the ion chromatography, OCEC, metals, Professor Astogi was talking about it yesterday. And then I'll take a portion of filter paper. I will do the INP measurement using cold stage, and a portion of the filter paper I will take for the direct extraction from the filter paper for biological entities. So this gives you much comprehensive information. However, if you are using any online instruments, that will give you the same targeted thing. Of course, it is having its own advantages. We will get the in-depth data. But if you are doing the offline characterization, it is having its own benefits. But of course, the challenges are also there, like which filter to use. So if you are having a fundamental knowledge on filters, what are the filter papers you are using? Quartz, Teflon, nuclear port. So if you want to do OCEC, what you will prefer? You don't have any preference. You can only do quartz, right? But if you want to do INP or bioaerosol, what you will prefer? Nuclear port. Because from nuclear port, it can be washed easily. The INP can be washed easily. DNA can be extracted. So there are a lot of, so we are also working on this problem, how to evolve with the same filter paper to get all these characteristics done. And then with sampler to use optimization to study the chemical, biological INP simultaneously. And of course, nuclear filters are good for INP, as I said, and quads are good for OCC analysis. So this is again a puzzle. But I think if you work and you are focusing on it, you'll be able to solve this. And you will second me in saying, yes, offline characterizations are also very, very good. So coming to the conclusion. So when we say conclusion, we are awake, right? So I'm proceeding towards the last slide, and we all know that there is an existing knowledge gap between aerosol cloud interactions. This is huge. It has already been said. So we should have more and more knowledge about CCA and INP aerosol cloud interactions to just solve the puzzle, piece by piece. So fundamental science questions on INP and bioresource remain unexplored. Challenges are like limited instrumentation facilities for offline measurements of INP, which I think now we are out of this challenge. And second is low to very low DNA concentration in aerosol samples. Because what we are trying to do is sometimes if we are collecting a lot of sample, then chemical characterization is not possible because your filter is overloaded, right? But if you are doing a diurnal sampling or suppose sampling in the clean environments like Himalayan region and all, you will get such a small amount that it will be very, very difficult to get this low amount of DNA extracted and to know the species level information, getting the ASVs and all. I got an opportunity to work in Pearl's lab during my postdoc tenure. He's a marine biologist, and with him I learned how to extract DNA and all of these things. However, I'm a chemist. So now we are good to go for this low to very low DNA also. So with all this, I think future perspective. Online and offline measurements of INP, precipitation, aerosols, and fresh water, because currently we are having limited understanding, limited data from India. As Professor Sachin has also said that that's why the predictions are not possible. So more and more data, comprehensive data will help us in answering the fundamental science questions. Direct extraction of whole genomic DNA followed by high throughput NGS should be followed, because if we are doing the culture, we are losing a lot of non-culturable bioaerosol species. So this is a preferred technique. And then increased spatial coverage of in situ field measurements, like we are having some pan-India plans to cover for the bioaerosol and INP. And yes, last but not the least, establishment of aerosol observatories at mid and high altitudes for long-term measurements. So with this, I close and the floor is open for questions. Thank you.
Any questions?
It is bioerosion, but it is bioerosion, but as a system or ion, there is a tendency to make precipitation contaminated. As I said, that single aerosol is very highly cause a severe health.
So what is your question?
This bioaerosol is good for
Oh, the question is bioaerosol is good or bad. So I ask you, aerosol is good or bad?
actually aerosol.
Bioaerosol is also both. So basically, basically when we are talking of mixed phase clouds, we are talking of ice nucleation, bioaerosol is having very important role. And apart from that, biogeochemical cycling, those, right? So bioaerosol is having a very, very crucial role in hydrological cycle and all of those. But of course, if you're talking about the human health thing, or of course the disease thing, or pathogenic effects, as he was mentioning, then it is bad. So it is like what you are dealing with and what you are focusing on.
Thank you, ma'am, for this nice talk. Ma'am, in one of your slides, you said that in winters, the biological INP are added. Means how they are added? It is due to the lower planetary boundary.
Oh, so you're saying how? So how is another question which we don't have answered. See, basically, this is the first study where we could do this, right? But I think after 5 years, if you ask me, we'll be having some answers on this. How? Because whenever we are talking of INP characteristics, or we are just, see, here we are just looking at the spectrum. Now, our next step should be that when we are doing the INP thing, we should do the chemical as well as biological characterization also to answer these how. So I think then only this how can be answered. I see 3 hands. Which one is speaking first?
First of all, thank you, ma'am, for this wonderful talk. My doubt is regarding the bioenergy. If I consider
Professor Sarji, your questions are coming to me, by the way. Yeah, but we work on biology. Go ahead.
if I consider for example, if I consider pollen grains, I have a very basic knowledge of biology, but I guess even for different plant species, the chemical composition and the proposition for pollens will be different. So will this chemical composition and the different proposition affect the CCN activity?
Of course. There is a very straight away answer to this. See, because if you're talking of bioaerosol, be it pollen, be it bacteria, whatever it is, right? In whatever composition it is, in whatever form it is going to affect its role and implications, whatever it is CCN or INP or cloud formation or whatever. So all these things are again going to, so it is like when we say the aerosol of Chennai is different than aerosol of Delhi and aerosol of Jammu. So similarly, the bioaerosol will also be different. It will be governed by different physical, chemical, and atmospheric dynamics and properties. So of course, the implications will be different.
When we can categorize pollen into further categories that can have different hygroscopic.
Yeah, so I don't know it because it's not my PhD problem. It's not my student's PhD problem. But next problem, if you focus on pollen only. So that's why I'm saying that if you want such deep answers, you have to really work hard, you have to focus on the problem, then only you can unveil these fundamental science questions. But it's a good question and so many such questions are there. So tomorrow, if you are planning your research, you can contact us.
Hello, ma'am. Am I audible?
Yes.
So, as both of you suggested or got the result that bioaerosols form mixed phase clouds even at the warmer temperature, right? Am I correct?
So these are not our results. These are the well-established facts which we also saw in our sample.
Yeah. So actually, this as aerosol cloud interaction is very uncertain process, and many of the studies suggest because of these mixed phase clouds, these cloud albedo is actually decreasing. So how this bioaerosols impacting this aerosol cloud interaction, and how we quantify that radiation, like effective radiative forcing of that.
You are again going to how? See, basically, that's what I am saying. See, at present, we have reached to a level. So again, if you talk of this kind of studies, right? In India, I think after Typex or something, we are not having any, aircraft studies, or we are taking samples directly from the cloud. And we are, studying these characteristics because I showed you the limitations. When we are studying INP or we are studying the bioaerosol INP spectra, we are doing on the cold stage instrument offline characterization, and there are certain limitations due to secondary and all of that. So if you want to answer those questions, of course, you have to have a comprehensive campaign where you can target, of course, taking some samples direct from cloud. Whatever.
One more question about this cold stage instrumentation. Can we, I don't know what is the dimension or size of your instrument.
Very small.
So can we like install this instrument in a weather balloon at high altitude site, and then we can measure those?
See, weather balloon actually we are also
that balloon, because whether it is a normal weather balloon, then it will fly and
no. But the problem is we are having a nitrogen cylinder also.
Yeah, that's it.
So, ma'am, you told that INP is not affected by urban and non-urban environmental polluted, non-polluted. Will we expect a change in INP based on this spatial extent, or if some regions have similar areas of chemistry, will we see?
See, it will definitely vary on the spatial extent. That is for sure. What I have, and I want to repeat this. See, whatever we have found in our study is one-time sampling which we did in India, right? And we saw that pollution is not affecting. If we are taking more number of samples, maybe the results are different tomorrow. But at present, we were confident because Chen et al. In China also, because China is also known for pollution. They also found that, okay, the pollution is not affecting the eye spectra too much. So the first thing is you should be very clear and you should stick to the facts that I am saying pollution is not affecting. Second thing is, if you're saying urban or rural, or you're saying from one location to another, of course it is going to vary because there are so many factors which are going to affect the ice creation characteristics.
So, ma'am, as these bioaerosols have this INP, so can they be used for cloud seeding in comparison to the conventional things which we use?
See, in India, people are not using, but some people must have tried, I don't know, outside India. But of course, as these are the ice nucleates, right? So they can be used. I don't have any knowledge about it as being used or not. But of course, and when you are saying bioaerosol is having INP, I'm not saying that bioaerosol is having INP. Bioaerosol is acting as INP itself.
We will stop here now, please. You can further discuss during tea break.
Yeah, so thank you once again. It was a total pleasure to be here and very thankful to the organizers. And thank you all for the patience.
Thank you, ma'am, for the informative lecture. Few announcements before we break for tea. Please submit your TATA forms today itself before leaving the institute premises. Those who want prints, please mail the documents to desk admin. They will print out. And then please be back by 11:45 for your assessment test.
I just want to thank both the speakers, Professor Sachin Gunte and Dr. Shweta Yadav, for kindly accepting our invitation and giving interesting lectures. And I think that I could see that there are a lot of interactions, so we can continue to do during our tea break also. Okay, thank you.
I just have one small announcement to make.
Yeah.
I want to take this opportunity to announce our with the school on aerosol measurements. Actually, it's at IIT Madras. Why this is not opening? So it's already open. So just try to visit this. I don't know where it has gone. Yeah, okay. So just want to do some marketing. That's it, nothing else. Thank you.
We can go for tea. We go for tea. Sure. They'll have an exam.
Sorry.